I will welcome to the stage our next team from Tulane University, Instapath.
By show of hands, who here has been affected by cancer or knows somebody who has cancer? Every year, 14 million new patients will find out they have cancer. On top of this, cancer is the second leading cause of death in the world. For most cancers, patients undergo biopsy procedures to get a definitive diagnosis. If the biopsy results shows cancer is present, surgery is usually the next step to remove cancerous tissue. The goal of biopsy is to obtain tissue that's representative of the potentially cancerous area. Non-diagnostic biopsies occur when the needle misses that area. The goal of surgery is to remove all the cancerous tissue. Positive margins occur when cancer is on the edge of that tissue. This means some of the cancer was left behind in the patient. Unfortunately, non-diagnostic biopsies and positive margins often lead to painful repeat procedures. You can imagine the stress and anxiety you have to go through for this type of procedure. I am Mei Wang.
I'm David Tolman. We are Instapath, a team of PhD candidates and faculty from Tulane University. Our first hypothesis was that physicians want to reduce their repeat procedures, and therefore they would value having a tool to identify non-diagnostic biopsies and positive margins. We tested this hypothesis by interviewing over 50 physicians that treat cancer patients. And these physicians are pathologists, radiologists, surgeons, and oncologists. We were surprised to discover that surgeons and oncologists do not value having a tool to identify positive margins. We thought that if we could give them a tool to identify positive margins, they could reduce their repeat surgeries. But they told us that most of the time, even if they knew that the positive margin existed, they couldn't locate it within the cancer patient. So they would not be able to do anything about it during the surgery. We did validate that radiologists, especially interventional radiologists, and pathologists value identifying non-diagnostic biopsies. They told us that if they had a good way to identify non-diagnostic biopsies, they could reduce repeat procedures. Over 5 million patients get biopsies every year in the US, and that costs over $8 billion. Now, 1 million of these biopsies come back as non-diagnostic. The current method to identify non-diagnostic biopsies is rapid on-site evaluation, or ROSE. Our next hypothesis was that repeat biopsy procedures happen when ROSE is inaccurate. So we went back to our potential customers to test this hypothesis. And they told us ROSE is supposed to ensure biopsy quality for a diagnosis. But the current technology is slow, destructive to the tissue, and the physicians can't see the whole sample. We validated our hypothesis. The physicians told us they're frustrated because they're using technology that was invented over 100 years ago. So this is what the current technology is like. Who knows what this is? How about now? Right? For the current ROSE, pathologists have to use this to guess the whole content of the sample. Now, sometimes they get lucky and get a good piece, but 20% of the time, ROSE fails. Because this is the Mona Lisa that pathologists use to diagnose cancer. Yet the current technology only allows them to review a small portion of the biopsy.
So we hypothesized if we can show the whole biopsy, the pathologists will be able to increase their accuracy for ROSE. We tested this hypothesis by interviewing pathologists. So all of the pathologists told us they could make more accurate evaluation if they can see the whole sample. So we got grant funding to build our first prototype. Here is how it works. We stain the sample with a fluorescent stain, place it on a slide, then take an image of the entire tissue. The pathologist can use this image for evaluation. We tested our system on over 83 human samples and published our data. We hypothesized the pathologists will love this because they finally get to see the entire sample. We tested by showing our prototype to the pathologists we interviewed before. Then we showed our prototype to 50 more physicians. But they told us they like the fact they can see the whole sample, but they didn't like the black and white image. They said they want a colored image that looks like what they're used to. So we then hypothesized we have to simulate the appearance of the histology slide to provide value for the pathologist. To test this, we demoed we pivoted to a different technology. It took us a lot of research and multiple iterations. We finally got a colored image. So we tested our prototype on 103 human tissues and published our data. Then we went back to our pathologists. Now they told us they love it and they will use it.
So now we've delivered our value propositions to the pathologists. And we thought that these value propositions would be the most important ones for interventional radiologists as well. To validate this hypothesis, we interviewed and observed 38 interventional radiologists performing biopsies. But they described their main pain point with biopsies is delays caused by rows. To our surprise, rows doubles their procedure time. So we hypothesized that interventional radiologists would pay for new technology to reduce procedure time. The interventional radiologists told us the reason why they would pay for it, and that's because they can treat more patients sooner. So not only did we validate that they would pay, but most interventional radiologists said they would pay $250,000 just to reduce the time they spend performing their biopsies. The interventional radiologists told us to focus on the kidney and lung biopsy market because these have the lowest Rose accuracy and the highest complication rate. These complications include excessive bleeding, which increases for every needle pass, anesthesia events that could stop breathing and lead to hospitalization, and a collapsed lung 20% of the time. So as you can see, it took a long time, and we stumbled validating our value propositions. But we finally validated them for both interventional radiologists and pathologists.
Our next hypothesis is that the physicians make purchasing decisions by themselves. And the best way we can reach them is through trade shows, conference presentations, and one-on-one demonstrations. So we interviewed 62 new physicians to test our hypothesis. We validated these channels are the best ways to reach our physicians. But we discovered hospital purchasing is more complicated. Most hospitals have a value-added committee, or VAC. They get together periodically to decide on what to buy. We want to quantify the value we can provide for the VAC. We can reduce the time of a biopsy procedure from 1 hour to 30 minutes. The hospital can double the number of billable procedures each day, which means at least $2,000 more per hour. At this point, we have a good understanding of our value propositions for each customer segment. The next step is to validate how to put our product into our customers' hands. We thought we can use a distributor, and that's the most effective way to reach our customers. We also thought our main revenue stream will be selling hardware for over $250,000. So we talked to 50 industry experts to test our hypotheses. After talking to potential distribution partners and small medical device companies, we find out the distributor takes 50% cut of the price, but they are not experts are selling smaller companies' product. After talking to the competitors, we learned they price their hardware at $100,000 range and generate recurring revenue through software licensing and disposables. We then talked to the VAC. They told us the device that's more than $100,000 per unit can take 18 months for them to make purchasing decisions because more people have to be involved. With this knowledge, we decided to pivot to a different channel and pricing strategy. So for the first few years, our team will be the direct sales team to drive adoption. Our product contains 3 components: the disposable staining kit, the hardware for imaging, and software for remote evaluation. We decided to price our hardware at $100,000 and generate recurring revenue through disposables and software licensing. At this price point, a sale will take less than 6 months to go through.
All right, that is time. We now have 10 minutes for Q&A with the judges.
Thanks for a great presentation. I'm curious why a lot of times when I work with startups, I advise them to focus on one particular target market or customer, and you guys have 2, pathologists and IR. Can you tell me about your decision to go for 2 versus focus on 1?
It's because of the way the workflow of biopsy evaluation, and they're linked. So we're solving one problem, and that's for biopsy evaluation. The interventional radiologists perform the procedures, and the pathologists make the evaluation.
Well, the other thing is I'm a broken record here. Competition, like other people must know that Rose is antiquated. So who else is kind of trying to make this better?
Yeah, so there are a few companies that are trying to do the same thing, but our main advantage is we are a lot faster, and our images is higher resolution than most of them. And even with the one company that's both fast and then has good resolution, they are specialized in breast because they can pick up a specific chemical marker that only present in breast.
Time to market, I imagine it's long, but am I right or wrong on that? So walk me through kind of time to market.
So we're going to be a 510K Class 2 medical device, but we will need to do some clinical trials to get that approval. It will take us 2 years to conduct that clinical testing. We need to spend 1 year transitioning our lab prototype into a clinical ready prototype, and then we'll be able to do the clinical trials, which will get us FDA clearance. So 3 years.
So tell us about how much of the market your product could represent. We saw the slide with how many biopsies are performed, but you're not the whole biopsy solution, right?
Yes, so there are about 1,675 hospitals in the U.S. that have the interventional radiologists to perform their biopsy procedures, and there are 5 million biopsies. That's our full potential, and we're focusing on lung and kidney, which is about 2 million. And they are depends on the hospital, they range about 1,000 to 3,000 procedures per year per hospital. And then there are about 100 high volume centers, and we hypothesize they need 3 to 12 units because there are some centers like Memorial Sloan Kettering that have a lot of satellite clinics, and they will need one for every clinic.
With our pricing strategy, that represents between $500 million and $1 billion of the market, of the revenue potential.
From like a unit economics basis, like how much is a biopsy and how much does your individual procedure cost?
Right, so a biopsy is, a hospital will typically charge a patient $2,000 for a biopsy. Now, what it's reimbursed by health insurance, that's a whole nother conversation. So if the biopsy is, if they charge $2,000 for it, our disposable component costs $75. So that would be an added cost on a per volume basis. But there is an upfront capital expenditure on our hardware component. But they would be able to recoup that based on the increased procedural throughput they can have because we can decrease the procedure time.
Okay, thank you.
Well, can you talk about the margins on each of these things? So the hardware, disposable software?
Mm-hmm.
Yeah, so the COGS for
Positive margins, right? Sorry.
That's
That joke.
The COGS for our hardware is about $20,000 per unit. That includes the components and also the manufacturing cost. And for our disposables, it's actually about less than $1 because the disposable is made with plastic and then chemical, which is very inexpensive. And the software is, as you know, is R&D one-time cost, and then once you have the software going, you just maintain it with one person. That will be it. So margin is about 80% for the hardware and 90-something% for the disposables.
And the status, you guys have a proof of concept of all this working already, or what's the status of?
That's correct. We have been conducting clinical testing. So we did a set of patient samples on biopsies, and I mentioned the margins earlier. So that was over 80 human samples, and then after we pivoted to the staining solution to get the colored image, we've now done over 100 clinical samples, and that continues to be ongoing, and we have new studies starting up as soon as we get back to New Orleans as well.
So how many clinics could buy the $100,000 piece of hardware in the U.S. roughly?
The 1,600. That's the maximum.
1,600.
Yes.
Yeah, but some institutions, because of the volume and the number of procedural rooms that they have, they may be able to buy more. For example, Memorial Sloan Kettering in New York City has the largest volume of biopsy procedures done by their interventional radiology department. So we anticipate that they could buy about 20 of these just based on the number of locations they have in New York and the number of rooms they have going on a given day.
Can you go back to your Canvas slide real quick?
Sure.
So, can you tell us about? I guess the recuse complication is because just fewer IRs that they're doing.
It's because of the reduced time of the procedure. So in the current biopsy procedure, it takes about an hour because the current rows, just all the physical steps and all the personnel they need in the room, is actually the longest part of the procedure. It doesn't take that long to get the tissue. So if you're reducing the amount of time a needle is sticking in a patient and reducing the amount of time they're under anesthesia, that's going to reduce complications.
So did you quantify how much quicker yours is? Like what?
Yes, we did.
Oh, sorry.
It is, so a current procedure will take an hour, and because of the time it takes for us to process an image, it's 30 minutes. So it's half the time it normally takes.
All right, that looks like all of the questions from the judges.
Thank you.